वक्ता तो नहीं मैं कहूँगा, बल्कि हमारे टीचर डॉक्टर सुनील कुमार गुप्ता जी, ए वेटरन, आई वुड से एनाटोमिस्ट, and I have invited, in fact, on behalf of Bihar Animal Sciences University, I had requested him to talk to the students, and he preferred to talk to them in a histological language. So, Dr. Gupta ji का बहुत बहुत स्वागत है. हमारे colleagues, Dr. Sanjay, Dr. Gautam, Dr. Manoj, and dear students, I think this is a great opportunity for us to interact with the teachers who are teaching in abroad for many years. डॉक्टर गुप्ता जी हमारे काफी लंबे समय से बाहर ही हैं, कई कंट्रीज में टीचिंग कर रहे हैं सिंस 1993 आई थिंक, इफ आई एम नॉट रॉन्ग. तो डॉक्टर साहब 94 से, तो डॉक्टर साहब काफी समय से वहाँ पढ़ा रहे हैं, और वहाँ का एटमॉस्फेयर क्या है टीचिंग का, उसके बारे में शायद ब्रीफ में डॉक्टर साहब थोड़ा बात करेंगे, और आपके साथ मेनली हिस्टोलॉजी पे फोकस करेंगे कि हर एक टीचर का एक अपना पेट टॉपिक होता है. तो डॉक्टर साहब डॉक्टर गुप्ता preferred to talk on that. So without taking much of the time, I once again welcome him for this talk, and so thank you very much once again. Welcome to the talk, Dr. Bharti.
Thank you so much, sir. Sir, we have with us total about 55 participants, and when they will appear, we will permit to join. So without taking delay, I would like to request and invite our today's speakers and guest, Dr. Sunil Kumar Gupta, sir. So please, sir, we will come with our topic.
Thank you very much, and namaste to all of you. Let me first share screen. Okay. Everyone hear me clearly?
Yes, sir.
Okay. Or you want a little louder? Let's check how much is
fine.
Yeah,
sir,
it's fine. I think it's fine. Okay. So let's start with let's first do the slideshow. Okay. That's my brief introduction, although Dr. Ravinder Kumar has covered almost everything. I'm still Indian national, Indian citizen, but resident in Grenada, and did my primary education. I was born and raised up in Jhansi, Uttar Pradesh, and did my veterinary education, my BBSc in 1982 and MBSc in 1984. And since then, I'm working as a faculty, several universities listed here, but most of these are like Sere Kashmir, only 1 year, Gujarat Agriculture University, 1.5 years, Himachal Pradesh Agriculture University with Dr. Ravinder Kumar, 7.5 years approximately. And so this was all up to 94. Then I went to University of Zambia, where I worked for 2 years. Then University of Zimbabwe for 3 years. And now the longest is the St. George's University of Grenada, completed 22 years. So initially all those 1 year, 2 year, and finally is almost settled here in St. George's University, Grenada. So first thing is you would like to know what the Grenada is. Grenada is a very small country. Very small country means is only 1 lakh population. We call it 100,000 or only 1 lakh. So compared to big cities like Patna, which is a city, not the country. This is like I was just talking with your Dr. Ravinder Kumar. He was telling 25 lakhs is the population of Patna, and this is only 1 lakh, and this has a status of a country. So it's a very small island, but is beautiful. Is famous for they have world famous beaches. So their main revenue comes from the tourism. And you might have heard about this Caribbean or West Indies. So West Indies world is more famous in India regarding the cricket matches. So I remember in 2008 when there was a World Cup. So although Grenada is a small island, but they have a world-class cricket stadium. So we have like 6 matches was held in 2008 World Cup, Cricket World Cup. So this is a, and okay, so advantage here is a pollution wise, health wise, very good. And with 100,000 or 1 lakh population, since since last March 2020, this COVID-19, we have only 165 cases only in last 1.5 years altogether, the COVID cases. And now we hardly has 1 or 2 active cases. So mostly they are recovered. So that's the reason we feel it quite safe. We got stuck here because a small island, we cannot go anywhere. All flights are cancelled. So this is safe. People are not coming here, not going out. So it's a very safe place as far as this COVID is concerned. Okay, so that's about island. That's my university, St. George's University. This university is famous for School of Medicine. It's famous for School of Veterinary Medicine and School of Veterinary Medicine. And you will be surprised to know that School of Medicine has almost 1,100 or 1,200 students twice a year. Like let's say 1,200 students coming in August, and then another 1,200 students coming in January. So one of the biggest as far as the number of students are concerned here. This is a American offshore university. You might have heard about, like in India, we have a lot of private university. So this is a private university, American offshore. Those who don't get admission in America, Canada, or we have like students all over the world, from Europe, Australia, everywhere, from India also, very few students from India also. So School of Medicine here was started in 1976, whereas the School of Veterinary Medicine started in 1999. So I was one of the first faculty to join the School of Veterinary Medicine here. And School of Veterinary Medicine, we are still restricted to the number 120, but 2 times in a year. So altogether, every year we produce 250, 240 students. They are produced every year. This School of Veterinary Medicine is accredited by AVMA. So that means is the American Veterinary Medical Association. So we are at par with any American veterinary school. So that is the reason why we get our, let's say 90% of the students here are either from US and very few from Canada. Then Europe and some local students also, Caribbean students also come here. So that's about the university. Most of the facilities are very much the same because I keep updating from like, I was looking at one of the posts from Dr. Ravinder Kumar showing the anatomies table. So we purchase anatomies and your university, your veterinary school also has the anatomies. So lecture room is very much the same. Our class is little different. We have 75%. Let's say out of 120, 90, 75%, or in some classes, even more than hundreds are the females. So a lot of female, you can look in this picture also. So in US also, the same trend in the American veterinary school also, they have 75%. So it's very much most of the facilities, because now the technology is so advanced everywhere. I know when I left Palampur and went abroad, we started all this PowerPoint, etc. But within a year or two, most of the places also. So right now there is really not really much different. That's my histology lab. And you can see like here, this is the stage and this has a microscope. I put the slide here and it goes to all these 6, 7 monitors. And these microscope, this is how the students are studying. So that's about histology lab. And one more thing here is what I has some difference here, like I'm not sure right now, maybe in India right now, because when we entered in BVS degree program, our average was 19 or 20 years, the average student entering in the class. Here, the average student entering the class age is 23 or 24. So this is they are a little more mature as compared to. Okay, so now coming to the main topic. So before I start the main topic, let me clarify here. This is exactly what I teach here in St. George's University, Grenada. So there is this is the same my PowerPoint which I use here. So nothing, just showing that this is what we teach here. Okay, so blood, as you already know, is a main purpose function of the blood is transportation and thermoregulation. So now is mainly this lecture is mainly for the students. Okay. Now, as every one of you knows that our body is having the cells, the unit structure of the body is the cells, and each and every cell of the body is a live cell. So they need oxygen and they need food to perform various functions. How they get the oxygen and the food? The purpose, the transportation of the oxygen and the food is by means of blood. So look at here. This is the oxygenated blood, or you can say what you inhale. When you inhale, the oxygen goes in the lungs. So from the lungs, it goes to the pulmonary vein, and from pulmonary vein, it goes to the left atrium to the left ventricle. From left ventricle through the aorta, it supplies throughout the body. Like if I have my hand here, all these cells, every whole body is composed of the cells, and below the cells is surrounded by a network of capillary. So here, this aorta is supplying the blood throughout the body, and this is a network. So network of the blood vessel, which is a capillary here, that is where the exchange occurs. So what oxygen you inhale, what food you have eaten is absorbed again in the blood, and all this is going in capillary. And in between the capillary, there is a fluid. So there is a circulation of the fluid is also there. And that circulation of that fluid is called lymph. So this is a lymph, and this is going through, and again, lymph vessels like biggest lymph vessel, thoracic duct, they again open in the venous side, and here the deoxygenated blood, excretory product, they are also coming in the right atrium. From right atrium, it goes to the right ventricle, and now going to the lungs again, where they release the carbon dioxide and they take oxygen. So this is again where the exchange of the gases occur in the lungs here. So just going a little more in the cellular level, this is actually the enlargement of this part here, this lower part. Look at here. This is the arterial side bringing the food and the oxygen. And this is all the capillary plexus is between the artery and the venous side. More specific, arteriole, venule, and this is the blood capillary. In between the blood capillary, you see there is a tissue fluid, and these are the cells. If it is a skin, you see these are the, if it is a liver, is a hepatocyte. So the name of these cells changes. So this is how the food and oxygen coming from the arteriole, coming in the capillary, where the exchange occurs. Look at oxygen and food coming out and then going back again and coming through the venous side. And but this fluid also need to be circulated. There's no stagnation. So this goes through the lymph capillary. Look at here, another picture. This is the artery with the red arrow. This is bringing the oxygenated blood as well as food. Look at here. And this is the capillary. This line here, this is the capillary. That is where the exchange occurs. That is where the oxygen and food coming out from the capillary, going in the cell. This because every living cell need oxygen. Every living cell need food. So they utilize the oxygen, they utilize the food, that at the same time they excrete the waste product here. So waste product, carbon dioxide also, and that is going back to the venous side. So this is the main blood circulation. But at the same time, everything coming in and going out by means of the tissue fluid. There is a fluid here, so this coming through the tissue fluid. So this tissue fluid also has to be circulated. So this tissue fluid goes through the lymph capillary, then lymphatic, different size of the lymphatic, and in the course of the lymphatic there is a lymph node. Very important point actually here, because what happens when there is an infection coming in the blood, how it goes in the tissue fluid, it can escape the blood vessel, goes in the tissue fluid, and then it comes in the lymph capillary, and this lymph node is all closely packed with the lymphocyte and macrophage. You might have done those histology of the lymph node. So this is how they take care of if there is a microorganism entering in the lymph vessel, and if it is still in the blood microorganism, then spleen take care of the blood-borne antigen, and tissue fluid, this lymph node take care of the tissue fluid, the one which escape in the tissue fluid. So this picture is explaining the basic of how the exchange of the fluid, through the fluid, oxygen and food through the capillary network, as well as the circulation of lymph occur. Okay, now what is the blood? Blood has mainly, these are the blood cells, three types of the blood cells: erythrocyte, red blood cells, thrombocyte, platelets, and the third is leukocyte. And then the remaining is the plasma. Normally the plasma is 8 to 11% in the large animal and 6 to 7% in the small animal, or you can say lab animal. So when you are centrifuging here, this upper part, this is almost like a 54-55%. This is the plasma here. And the lowest part here, this is called packed cell volume. This is mainly the erythrocyte here. And in between a buffy coat, that actually represent the white blood cell. So this white blood cell is almost, you can say the thrombocyte and leukocyte is only 1%. So this is about, okay, so that's the erythrocyte, one of the most beautiful cells of the body. Look at here, but they are non-nucleated biconcave disc in most of the domestic animal. Initially they are nucleated, but in the process of maturation, they lose their nucleus, they lose their cell organelles. Here there are some species variation. In goat and pig is a flattened disc. Now here this is also some species variation. Normally the dog has the largest erythrocyte and goat has the smallest. Although there is this is just a species specific. So don't ask me why the dog has largest. So it doesn't have any physiological, not much, but this is mainly the species specific. Okay, so it's always good to have some correlation. We call it anemia. Anemia means when the red cell measure by RBC count, you might know like 1 ml of the blood has a limited number of the red blood cells. The number can increase or decrease, so its number is decreased. Then hemoglobin concentration or the packed cell volume, any of these or all three of these, this is when there's a decrease in these parameters, then we call it anemia. So anemia is quite a wide term here. Reticulocyte, important because you see that in some of the disease condition, reticulocyte is an immature RBC. When they are immature, you still see some DNA material here and some ribosomal, because during the process of maturation, most of these, they lost their nucleus, they lost their ribosomal RNA, but some still remain. So these are the immature RBCs called reticulocyte. Now here, whole jolly bodies. This is a DNA fragment, as I was mentioning here. Originally, these when they are developing, RBC are developing, they have a nucleated, and in the process of maturation, they lose their nucleus. So nucleus is mainly the DNA material, and here some of the DNA are still remain there. So up to certain level, this is a normal, but after that is not. So these are called Howell-Jolly bodies. So that's the. Now Haynes bodies, these are like a oxidized hemoglobin or denatured. As we were mentioning, the main purpose of the erythrocyte or the hemoglobin is to carry the oxygen. They join with oxygen, they carrying the oxygen, release in the tissue fluid, and finally to the cell. So these are some of the hemoglobin which is denatured, which is oxidized, which is not useful. So this is although 2 to 3% of Haynes bodies are normal in the cats, and this is why most of you might be aware that normally we avoid the raw onion, keeping the raw onion in front of a dog and cat, especially in the cat. That is not the safe. This is what this is causing this toxicity. So just avoid keeping a raw onion in front of the cat. So otherwise 1 to 2% is normal, but after that it is pathological. So this is the Haynes bodies. Rolu formation. Rolu formation, when the blood is inside the blood vessel, is flowing, is perfectly all right. When it comes out, then these RBC, they start adhering each other. They start joining with sticking with each other. They form like a stick of the coin. Look at here, like you put the coin one after other. So this is phenomena is called Rolu formation. And this Rolu formation is the most prominent in horses and cats. Is not as prominent in dog and pig, and is rare in ruminant. So that's Rolu formation here. This RBC, when you are looking in the slide in the histological lab, you see here, because it's a biconcave. Biconcave means the edges are thick and center is thin. So when you are looking that in the slide, this is why you see the paler. When there is a thin or the thick one is always the darkly stained. So all these other than this we will discuss a little later. So this is these are the RBCs here. And RBC has 33% of the hemoglobin. So main role of the RBC is this hemoglobin is to carry the oxygen, and it's distributing throughout the body. All cells need oxygen. So when they are developing, they have the nucleus, then they lose all these mitochondria, ribosome, and many cytoplasmic enzyme during maturation. Now sometimes students ask, so if they lose everything, so that is why they have a limited lifespan. They don't live long. So in the process of maturation, they lose everything, and their main role is related with the hemoglobin. So they don't really need a lot of activity here. So this is different animal has a different lifespan. So that is mentioned here. Now coming to the white blood cell. White blood cell altogether is called leukocyte. There are two types. One is called the polymorphonuclear granulocyte, and other is called mononuclear agranulocyte. So this granulocyte and agranulocyte is based on the presence of specific granule, and those specific granule, when they stay neutral, then this is called cell is called neutrophil. When they stay acidic or eosinophilic, then it is called eosinophil. When they stay basic, that is called basophil. So these are the three types of the granulocyte. So, and they are polymorphonuclear. So their nucleus is multilobed. Now this is the mononuclear agranulocyte. Although it says agranulocyte, means they don't have these specific granules. Then sometimes students say that because you mentioned in the notes that they have a xerophilic granule. So it's not that they don't have a granule, they have granules, but those are linked with the lysosome. If you have done this lysosome in your cytology, you might have knowing that they have the hydrolytic enzyme. These xerophilic granules, they are hydrolytic. They are not only present in agranulocyte, they are present in all five. So, but these specific granule by which these leukocytes are divided, they are only present in granulocyte. This is why we define granulocyte and non-granulocyte or agranulocyte based on the specific granule, which has the staining either neutral, acidic, or basic. Okay, here the differential leukocyte count. Picture table looks quite big, but easier to remember. Like basophil is normally 1%, eosinophil 2 to 5%, very little, much variation in the normal monocyte also. Now here, very interesting thing here. Look at the horse and the dog and cat. These three, I consider those as a pet. So dog, cat, and horse. They have more than 50% of the neutrophils are there. Like in horses, 50 to 60%, and dog 65 to 70%, and cat 50 to 60%. Now remaining all animal, you look at here, pig, cow, sheep, goat, and chicken. Their lymphocyte is more than 50% is lymphocyte. So the point to note here, look at this dog. Dog has 65 to 70% of the neutrophil in dog is perfectly normal. But if you have 65 to 70% of the neutrophil in cow, this is very pathological. So something here like neutrophil 65, 70%, or in cat more than 50%, horse, something which is normal in horse, dog, and cat, but that is a pathological, or you can say acute disease, because 65%, if you see in case of the sheep and goat, let's say if sheep or goat has 65 to 70% of neutrophil, is highly pathological. Similarly here, look at in pig, lymphocyte is more than 50%, and here is only 30 to 40%. So this second category of these animals where the lymphocyte predominate. So this is the pig, cow, sheep, and here is the chicken and goat. So these are, so there are two categories of animal. One has predominance of lymphocyte, one has a predominance of neutrophil. Okay, now coming here one by one, discussing about all neutrophil. This is their size, and these are multilobulated. We already mentioned segmented nuclei, 3 to 4 lobes, and this is a bar body. This, although bar body is prominent in neutrophil, this is how only present in the females. If you guys remember, you might knowing that XX, there are 2 X in female and XY in male. So the second X is actually considered as a bar body, which is not really a very active chromosome, but is important, especially in the neutrophil, and you can recognize that this blood smear belong to male or female. Okay, cytoplasm is pale, means light staining, because their granules are neutral. They are neither acidic nor basic. Very important, this is a very important cell. They have 2 types of the granules. One is a specific granules are bactericidal. They kill the bacteria and then they do their hydrolysis also. They digest the dead part. So this is the important, that not all other cells has these specific characteristic. They have bactericidal as well as they have the hydrolytic, like here. Okay, one point here is a band cell. You might do that in pathology, or nowadays everything is correlated. So band cell is an immature neutrophil. Like here, you see this is band, is a U band or U shaped. So you don't really see this segmented here. So these are, you can say these are the band cell, immature neutrophil. Is important because in some disease condition, the number of band cell increases. So these are the band cells here. These are the, how they work. Look at here, this is inside their blood vessels, and they don't live actually long. Most of these WBC, they only live 3 to 10 days. But here, when they come in circulation, then this neutrophil is only 10 to 14 hours. how it reach, like here, this is the blood vessel. The lining of blood vessel is the endothelial cells, and here is the tissue. So if you have a injury or you have a microorganism which develop here, so these they actually normally they are moving on the side wall of the blood vessel, like by rolling, and then they stop. They get chemotaxis by the smell, chemical release, and they just going through they transform, they're migrating in the tissue space. And look at here, they are not only eating, they are digesting also. So this is how these phagocytosis, their function is called phagocytosis. They kill it, they have bactericidal as well as they have hydrolytic enzymes. This is how they work here. Okay, now the second one is eosinophil. As its name say, those specific granules they have is eosinophilic. This is why this is called eosinophils. And normally the nucleus is bilobe here, and is very important actually. It secretes the antihistamine, or you can say this inactivate the histamine. Histamine is normally secreted. If you remember, in the connective tissue, there's a mast cell which secrete the histamine, whereas in blood, the next we are going to discuss, which is a basophil, which secrete. So this function of the eosinophil is to inactivate the histamine, because normally when you have an allergic reaction, in allergic reaction, basophil is secreting the histamine. So purpose of histamine is to dilate the blood vessel, to neutralize the effect of the microorganism or the allergen. But then the eosinophil help to inactivate, to bring it back to the normal. Then it also help in the killing the parasitic larvae. These eosinophil also help to kill the parasitic larvae. So this is the reason why, if you are seeing a number of eosinophil increased, like more than 2 to 5, whatever the normal range is, that means this blood smear, either they have the animal has the parasitic infestation or they have the allergic reaction. So number of eosinophil increases in the parasitic infestation as well as allergic reaction. As we are discussing some species difference here, the horse eosinophil is different than the other. You cannot differentiate all other, but you can differentiate if it is a eosinophil of horse. Their granules are very big, huge. And look at here, sometimes you hardly see the nucleus here. They are extremely large and tightly packed. So altogether, easier to find in the slide, because they looks like a mulberry-like appearance here. Okay, now the third of the granulocyte is the basophil, and you can look at these granules here. These granules are basophilic. So they also have a heterochromatic nucleus and metachromatic granules. Very much structure is same as the mast cell, if you remember, because mast cell also release the histamine. This is the histamine. Is a very important cell when there is an infection or inflammation occur. So inflammation, if it is caused by a microorganism, bacteria, then this release the histamine, which dilate the blood vessel to dilute the effect of the microorganism, or to dilute the effect of the toxin, to dilute the effect of the allergen. It also releases the heparin so that blood will not clot inside the blood vessel. Is an anticoagulant, heparin here. So this is like comparing all three. When you are identifying those in your slide, one is his name is very self-explanatory. One has a basophilic granule, eosinophilic granule, and neutral granule here. Now coming to the lymphocyte. Lymphocyte is a part of your immune system, big part of the immune system. These are, like you see, big nucleus, no lobes, that's why you say this is a small rim of the cytoplasm here. And this is classified under a granulocyte. There are 3 types of the lymphocyte. Very important to know. One is a B lymphocyte, second is the T lymphocyte, and the third is NK or natural killer cell. When I say B lymphocyte, why it is called B lymphocyte? Because these cells mature in bone marrow. So this is why B for bone marrow, because these lymphocytes mature in bone marrow. T lymphocyte, T for thymus. They need to go to the thymus to get the maturation, and this is why it is called. The third one is a very specific, which is called NK, natural killer cell. Histologically, you cannot differentiate. In your slide, you will see all lymphocytes same. This is how you differentiate. Okay. So how these are different? B lymphocyte. B lymphocyte, as I mentioned, they develop in the bone marrow, and these are the one which form the antibodies. This is the antibody, which is actually the basic of all these vaccine. In the vaccine, you insert the non-pathogenic antigen and to create the antibody. And this is the antibody. And let's see, this Y is an antibody, and these dots are antigen. So they trap the antigen. They neutralize the effect of the antigen here. So now the antigen is no longer functioning. So this is, they produce the B lymphocyte, they produce the antibody. Look at here, the plasma cell also do the same function. In the connective tissue, you might have done, there is a plasma cell. So those plasma cell is actually produced by the B lymphocyte. These cells, they convert into the plasma cell when they go in connective tissue. And they also actually, these B lymphocyte also develop some memory cell. Memory cell means once they remember that this is antigen, they remember. Next time, any antigen, or you can say enemy, or the coming, they recognize. They immediately form the antibody. And this antibody, antigen, is a basic of all your vaccination. Or so, but they are only 10% of the circulating lymphocyte, which forms the antibody. This is the plasma cell. You might have heard about the cartwheel-like appearance of the nucleus, eccentric nucleus. All this is, all these basophilic cytoplasm, they are derived from B lymphocyte. Okay, T lymphocyte. What is this T lymphocyte is doing? The first thing is T lymphocyte is about the 75%. They are the majority of the lymphocyte are T lymphocyte. And how they work? This is a, let's say, the infected cell. They just kill the infected cell. They perforate. They are like a shooter. They're just perforating here, the cell membrane. So, and then they kill the infected, lysis of the infected cell. So this is why we call it participate the cell mediated immunity. But they also has a memory cell. So they are also helpful. They are not forming the antibody, but if their antigen is coming, they are directly killing the antigen. We are directly killing the infected cell here. So this is the difference. One is making the perforation like a bullet gun, just making the perforation in the cell membrane and finally killing the infected cell. Whereas this one is making the antigen inactivate here, making the, because of the antibody, they form the antibody and antibody inactivate. They don't inactivate, they just kill here. The lysis of the, both of these has the memory cell. They remember their foreign body, they remember their enemy, what is coming, what the antigen, what the pathogen is. Now, third one is a NK cell, natural killer cell. Our body, most of the cell, let's say in the skin, some live 7 days, 10 days, 20 days. So there is a continuously cell division occur. Cells of our cell, most of the cells of the body, they are continuously dividing and dividing. When they are continuously dividing in the process of mitosis, some of the cell, there some genetic changes occur, and those are called the cancer cell. Cancer cells are present in everybody. Everyone, when your cells are mitotically active, they're actively multiplying. There's always some percentage, 1%, 2% of the cells, they are always converting into the cancer cell. But how we all are protected? This is the NK, natural killer cell. These NK cell, these are the one which killing those cancer cell and protecting all of us from the cancer here. Now the different, why this is different? They don't have the antigen receptor. They don't have memory cell. This is the reason we don't have the vaccine for the cancer. We have the NK killer cell, we have a natural protection for the cancer, but there's not yet, we never developed a vaccine, because these NK killer cell, which is also a type of the lymphocyte, this is the third type of lymphocyte, and this is providing protection against tumor cell or the cancer cell, but it lacks the antigen. This is what is called innate immune system. Innate means they don't recognize. As far as how they work very much like that of the T lymphocyte. So these, they also make the perforation. They also kill the infected cell. They see the cancer cell, they just kill. So this is 10 to 15% of the blood lymphocyte are the NK killer cells. So now we're coming back to all three. One is a B lymphocyte maturing in the bone marrow. They form the antibody. T lymphocyte, they just killing. They have some toxic materials to kill this, perforate the cell membrane and then kill. And natural killer cell, very specific, only working on cancer cell. So these two, they have a memory cell, they recognize, but the third one, natural killer cell, this is not recognized. That is why we never had the vaccine. So they don't have a memory cell. Now coming to the monocyte, which is the largest here, largest of the leukocyte. If you look at the size, almost like a 12 to 18, kidney shaped. Now here, this characteristic, they have the hydrolytic enzyme. So how you will, what is the significance here? This monocyte don't really has a bactericidal compound. So it cannot kill the microorganism. It can eat, then it can digest. Hydrolysis means digestion of the killed material. So this always need to work together with the lymphocyte. So lymphocyte is killing, whether it's a or inactivating, B lymphocyte inactivating, T lymphocyte killing, NK killer cell also killing. So what is that killed material or inactivate material is processed by these hydrolytic enzyme. They have the monocyte. So monocyte always work together with the lymphocyte. Look at here, the contact of lymphocyte and monocyte is necessary. Now these monocyte is in the blood. You might have done a lot of tissue macrophage. Let's say remembering the Kupffer cell in the liver, that's a phagocytic cell. There's one phagocytic cell in nervous tissue that is called microglial cell. One phagocytic cell in the lungs is called pulmonary macrophage. So all these phagocytic cells of the body, they are all derived from monocyte. So this is a monocyte in the blood. When it goes in the nervous system, it is named as microglial. When the same cell goes in the liver, it changes the name to Kupffer cell. So you can say is a precursor of all the tissue macrophages. In other words, you can say is a mother of all the phagocytic cell, or you can say the tissue macrophage, whether it's in the liver, whether it's in the lungs. So this is they do active, they have the lysosome, all these phagocytic cell, they are having the abundance of the lysosome, and lysosome is the one which actually has these azurophilic granule, which is a hydrolytic enzyme. Now coming to the thrombocyte or platelets, here these are smallest one, tiny here, and they have the adhesive property, they stick, they have microfilament, microtubule, and their main function is the coagulation of blood. So they do the blood coagulation. So smallest of these blood cell is a thrombocyte. Look at the size, is very small here. Okay, so now we are done with all type of the cells. So normally when I lecture, I have something like this to review. We discuss lymphocyte, 3 types of lymphocyte, and T cell, B cell, and NK. We discuss neutrophil. The basic feature of the neutrophil is to bactericidal as well as hydrolytic. We discuss the eosinophil, which is antihistaminic as well as anti-parasitic. RBC mainly carrying the oxygen, having the oxygenated or the hemoglobin. Basophil help in the inflammatory reaction, they secrete histamine and heparin also, and monocyte is a phagocytic cell. Okay, a little bit about avian. So the first characteristic of the avian blood is the RBC is nucleated. So this is how you differentiate whether this is a blood smear belong to mammal or blood smear belong to the avian. So this is all these are the. Another difference here is the thrombocyte is really big, as is almost like a nucleus of the RBC. So these are the thrombocyte. If you compare in mammal, they are really small. So relatively, this is big. Another important point here is the neutrophil. We don't call the neutrophil in avian blood. So here, instead of neutrophil, is called heterophil. So that's the difference. Rest, most of these are the same, monocyte and lymphocyte. So some of these differential feature in the birds or the avian blood here. Okay, so a little bit about the hematopoiesis or the how the blood is formed. Might have done in embryology. Originally, it is produced in the yolk sac, and then in the embryonic life, then in fetal life, these are produced in the liver, also liver, spleen also, and then they all coming in the bone marrow. So bone marrow are two type, red or yellow. One is active, one is inactive, but this changes with the physiological status. Active can be non-active, depending on the physiological. So all these cells, they develop in the bone marrow except the T lymphocyte. So T lymphocyte is developing in the thymus. So lymphocyte, mainly where the lymphocyte are present, mainly goes in the lymph node and spleen. So they are produced in the bone marrow, then T lymphocyte goes in the thymus, and then finally they go to the lymph node and the spleen. That is where they take care, they filter the lymph, or they take care of the antigen coming in the blood. So that will be the spleen, and if antigen coming through the tissue fluid, then this is the lymph node. It take care of this. So how these are going in the bone, most of you have done the study of the bone and may have noticed this nutrient foramen. So nutrient foramen is actually where the nutrient artery is going here. And this is carrying what the blood cells produce in the yolk sac and the liver, and coming through the blood circulation, coming in the bone marrow. Look at here, this is the nutrient arteries. You might have done those central artery, Haversian system, all those. And here, this is the marrow cavity or the medullary sinuses here. So this is where all the blood cells we were discussing, they are produced in the bone marrow here. So hemopoietic. Then they goes in the venous side, and now again going in the circulation. This is where they get maturation, where they are formed, originally coming from the liver or yolk sac. Some are produced in the bone marrow. They mature here, all the development, and then finally going back in the blood through the venous sinus. So this coming through the artery, going out in the circulation through the vein, and they are in the venous sinuses and then going. So all these cells, blood cells, they are produced and then going through the peripheral circulation here. Now, all these cells, if you might have done a cell in the connective tissue, and that is called mesenchymal cell, one of the very important cell, which is mainly in the embryonic tissue. And mesenchymal cell, look at here, this cell has a tendency to change in any type of, most of these cells are derived from mesenchymal cell. Most of the connective tissue cells, they are derived from mesenchymal cells here. So these mesenchymal cells, they developing, the one which is developing into the blood cell is called hemocytoblast. And these are the pluripotent cell. Pluripotent means plural. Pluri means plural, potent means capacity. They have capacity to change in the various types of cells. So here, they are in the blood. Mainly there are two types of the cells. One is lymphoid. This is a lymphoid system is completely different. So lymphoid system is developed into lymphocyte, NK, those natural killer cell for the cancer, T lymphocyte and B lymphocyte. And B lymphocyte further change to plasma cell. Rest all other cells is called myeloid system, myeloid stem cell, which is going in the erythrocyte, or this is the monocyte forming the macrophage. Then granulocyte, they convert into neutrophil, basophil, and eosinophil. So these are the three types of the granulocyte. Now here we look at the blood platelets. If you remember, is the smallest cell, but here this megakaryocyte looks biggest. So how this form the blood platelet? This is a bigger, like that is why it is called megakaryocyte. So these small pieces here, it actually releases the fragments. It is releasing a small fragment which becomes the platelet. So this help in the blood coagulation. So that is about the hematopoiesis, and that's all. If there is any question? Okay, so this point here, let's have a little more emphasizing here. Look at this one, especially like how the microorganism is traveling here, if it is coming in the blood, if it is still in the blood, and then finally it will go to the spleen, and the spleen is all packed with lymphocyte and macrophage. So when they're passing through the spleen filters the blood. But if the microorganism escape in the tissue fluid here, now it is going in the tissue fluid. Tissue fluid is marked as a green, which is called lymph. Now what is in the lymph node? Lymph node is again packed with those lymphocyte and macrophage. You can call plasma cell also, but B lymphocyte is same as plasma cell. So if the microorganism coming in the lymph, then this is a series of, let's say, thousands of the lymph node you have. If you want to feel like one below your mandible, this is the mandibular lymph node, parotid here. And in dog, you might have seen the prescapular lymph node. So these are this is what take care of the fluid borne, or if any, you have the microorganism in the fluid. So this is the basic of all the blood and lymph circulation. This is how it works. Okay, any question?
Any student have any questions?
Yeah, student or anyone can ask in Hindi also. आप हिंदी में भी पूछ सकते हैं.
Gaurav.
Yes, sir.
Any questions? कुछ भी अगर पूछना है सर से पूछो.
I think everything is clear, sir. It's very well understood.
Oh, okay. Okay. So how many girls you have in the class out of 55?
So 22 are girls. We have 59 students out of 22 are girls.
Okay, that's a good actually. When we were BBSC student, we didn't have any girls. So, but that was in early 80s. But when I was teaching in Himachal Pradesh, by the time I was leaving in 94, we have about 20%, 20-25%. So you might remember the quote that the development of a nation depends on how much the girls are educated there. So more like in America, US, we have 75% of the girls in the classroom. So that's a good, like you have 22 means almost like a 40%, 35-40% of girls in the veterinary medicine. So this is exactly what we teach. Although when I teach, it's almost the same. This is a 45-minute lecture, but I do a little more repetition. But I know that you guys have already done this part, so I might be a little fast here. In actual classroom, might be a little slower.
वॉल्यूम
And
थोड़ा सा कम करना.
सर एक क्वेश्चन है सर. नमस्ते
पहले
सर.
नाम बताएगा.
नमस्ते, नमस्ते.
भावेश कुमार सिंह 253 रोल नंबर सर.
Okay, yeah, Bhavesh Kumar, yes. So you have a question, Bhavesh?
यस सर.
Yes.
सर मैं सर ये जानना चाहता हूँ क्योंकि ब्लड
Yes.
हम लोग में सिक्रेटेड होता है और क्या कहते हैं हम लोग में होम्योस्टैटिक मैकेनिज्म पाया जाता है. तो सर फिर हम ये कैसे कह सकते हैं कि ब्लड का कंपोजिशन ऑलवेज हमारे बॉडी में सेम रहता है और अदर्स फिनोमेना में क्या कहते हैं, अगर कोई ड्रास्टिक या फिर कोई अगर स्मूथ कंडीशंस कोई आते हैं तो हाउ आवर बॉडी से हाउ आवर बॉडी tackle such drastic situation during abnormal.
Sorry, I don't understand. What is the?
Dr. Bharti, could you get the question?
भावेश,
यस सर.
क्या पूछा हमें भी नहीं पता चला, बीच में तुम रुक गए.
सर मैं ये जानना चाह रहा
अच्छा,
हूँ
आप
कि
अपना
सर
वीडियो तो खोलो यार, आपकी शक्ल
चेहरा दिखा भावेश.
जैसा शेयरिंग ऑफ कर देते हैं.
अच्छा, that's right.
अच्छा.
हाँ, बोलो भावेश, फिर से बोलो.
सर मैं ये कहना चाह रहा हूँ सर, हम लोग में तो ब्लड सिक्रेटेड होता है बाय द सेल्स.
हाँ.
और हम लोग में सर होम्योस्टैटिक मैकेनिज्म भी पाई जाती है.
यस.
तो सर हम लोग ये कहते हैं कि हम लोग का ब्लड का पीएच इतना, ब्लड में ये सब कुछ ये रहता है. तो हम लोग ये किस प्रकार से कह सकते हैं कि ब्लड का कंपोजिशन हमेशा नॉर्मल रहता है? हम लोग में क्या कहते हैं ब्लड सिक्रेटेड होता है. अगर मान लीजिए कि अगर किसी समय क्या कहते हैं सर हेड्स कंडीशन आता है, किसी समय क्या कहते हैं कि अनफेयर कंडीशंस आते हैं, तो मतलब ब्लड अपना होम्योस्टैटिक मैकेनिज्म के द्वारा अपना क्या कहते हैं ये मतलब जो सिचुएशंस होता है उसको हैंडल कर लेता है. तो फिर हम लोग का जो नॉर्मल फिजियोलॉजिकल मैकेनिज्म है वो कैसे संपन्न हो पाता है सर, मैं ये जानना चाहता था.
ये बॉडी में चेंजेस होते रहते हैं ना. जैसे अगर एलर्जी कंडीशन है तो इस्नोफिल बढ़ जाएगी, इन्फ्लेमेटरी कंडीशन है तो बेसोफिल बढ़ जाएगी, एनीमिया है तो ये कंडीशन, जिस बॉडी कंडीशंस के हिसाब से ब्लड का जो कंपोजिशन है, जो सेल्स हैं, वो बढ़ती घटती रहती हैं. तो what we discuss is actually the one for the normal conditions. जैसे कभी ऑक्सीजन लेवल कम हो रहा है तो हीमोग्लोबिन इफेक्ट कर रहा है, तो ये सारा जो सब लिंक्ड है. This is all linked with the pathological condition or the disease condition. So this is how these different cells.
तो फिर हम ये कैसे कह सकते हैं कि हमारी बॉडी हमेशा नॉर्मली हमारे बॉडी में सारे फंक्शंस नॉर्मली हैपेंड होते हैं? तब तो हम ये नहीं कह सकते हैं.
If everything is normal, if we are healthy,
hello sir,
if we are healthy, if we are normal, then everything is happening normally. So all these functions, everything is normal. But if there is although here there is something between the normal and when we say disease. Now what actually happens here when we get infection, all right, then our main army is a lymphocyte and macrophage, neutrophil. They are fighting with these microorganisms. So bacteria is multiplying, then our lymphocyte is also multiplying. So there is like a war-like condition. There is a war going on between the body defense mechanism and the microorganism. Some microorganism, they are releasing the chemicals. Some they inactivating the lymphocyte like the HIV virus. They destroy completely the lymphocyte. So they cannot, the immune system, they completely destroy. So what actually happened? Normally this war is going on always. Always we have microorganism, always some fight is going on. But when our defense mechanism fails, then we get the symptom. So that means you feel the fever. You get the symptom, and when you get the symptom, now you need antibiotic because your defense system cannot kill the enough microorganism. All right. So when your defense cannot kill, you need antibiotic to kill the microorganism. So this is how this blood and its defense mechanism, how its oxygen carrying capacity, food carrying capacity, so how this all work.
तो हम सर ये कह सकते हैं कि हमारा नॉर्मल फिजियोलॉजिकल मैकेनिज्म जो है वो क्या कहते हैं कि वो हमेशा मतलब बदलता रहता है अकॉर्डिंगली द सिचुएशन।
वो तो नॉर्मल में ही आएगा ना सारा, नॉर्मल तो नॉर्मल ही है। देयर आर टू कंडीशन, आइदर नॉर्मल और पैथोलॉजिकल। तो नॉर्मल थोड़ा बहुत तो वेरिएशन होता रहेगा, मतलब ये नहीं है कि तो व्हेन वी आर हेल्दी एवरीथिंग इज going well. When we are not healthy, and sometimes like not healthy means when we get the symptoms. So when our body defense mechanism is not that everyone looking healthy don't have the COVID. We might have, but our defense mechanism is strong enough so not to show any symptom. I think, but now most of you are, I hope most of you got vaccinated with COVID.
Yes, sir.
Okay,
yes,
we
yes,
also got vaccinated. Then only we are allowed to enter in our campus. So
तो सर हम ये कह सकते हैं कि अब हमारा जो सेल है।
डॉक्टर गुप्ता, आई थिंक मैं इनको डॉक्टर भवेश को समझाता हूँ थोड़ा फिजियोलॉजिकल लैंग्वेज। ही इज आस्किंग फिजियोलॉजिकल क्वेश्चन। भवेश, एक सिंपल एग्जांपल मैं देता हूँ आपको। से आर यू ए ब्लड डोनर?
यस सर।
आर यू ए ब्लड डोनर? ब्लड डोनेट किया है कभी आपने?
नो सर, आई हैव।
ओके, if you donate, whenever you donate the blood, some quantity of blood is taken out from your body. So when blood is taken out, there will be decrease in number of various cells in your body, and that results in influencing the erythropoietin. So blood formation begins, but for a shorter duration, the count of the different cells is low, but that helps in stimulating the blood formation mechanism, and that is how it is compensated. So this is how our body homeostatic mechanism operates. I think in a very simple way I have tried to explain you.
Yeah, very good.
Fine. और स्टूडेंट्स कोई क्वेश्चन पूछना चाहेंगे डॉक्टर साहब से?
निशा, have you any questions? Shreya.
Amit, you have any queries?
No sir.
Okay. चलिए, Dr. Bharti, go ahead if there are no more queries.
Okay, sir. Do you have any question, students? Last. Smriti Raj.
No, sir.
No. So let us we are near to conclude our today's e-lecture, sir. So in last I would like to request Dr. Ramesh Margatham for vote of thanks.
Thank you, sir. A very good evening, everyone.
It's actually my pleasure teaching in the school, in the veterinary college in India after so many years.
Thank you, sir. Very good evening to everyone, welcome. One and all. Today's our speaker, Dr. Sunil Gupta, sir, Professor and Course Director, Veterinary Histology and Embryology, St. George University, Grenada. Our respected Director Research, Sir, Bihar Animal Science University, Patna, and all the dear participants. It is great privilege for me to get opportunity for the vote of thanks for the today event. On behalf of the Bihar Animal Science University, Patna, I extend a warm welcome to all who joined in this platform. I would like to express my gratitude to all esteemed delegates of this webinar for their presence and contribution to make this webinar a great success. I also extend my great gratitude to our honorable chief guest, Dr. Sunil Gupta sir, to take out time from his busy schedule to grace the event. Thank you, sir, for inspiring and encouraging us with your beautiful words on this special day and adding such a beautiful knowledge regarding the different types of the blood cells. A very special thanks to our Honorable Vice Chancellor Sir, Director Research Sir, Dean of the Bihar Veterinary College Patna for providing immense support to make the webinar successful. I also extend my deep gratitude to Dr. S.K. Bharti Sir, Head Department of Veterinary Anatomy, to organize a webinar even in this pandemic situation. I must thank to all participants for their active participation. Thank you, everyone. Once again, thank you very much, sir.
Thank you for inviting me.
ठीक है सर, थैंक यू वेरी मच, थैंक्स अ लॉट.
Okay, namaste.
ओके सर, नमस्कार.
Lastly, I thank to Dr. Gautam sir for this beautiful presentation of vote of thanks.
All
Hope we will again be with a good touch with Dr. Sunil Gupta sir.
Thank you.
And we will arrange, sir, definitely. That was the very beautiful that has spoken by
Yeah,
Dr.
no problem. Anytime. This is I don't really has to work anything extra, because this is all my PowerPoint and what I teach here.
But sir, I have taken my degree from Pantnagar, Guwahati, and other also participated in the about 10 international national seminar anatomists. But your presentation was really nicely, sir.
Okay, thank you.
That was on the diagnostic approach, because we are only going towards the anatomical features.
नहीं, अभी प्रॉब्लम क्या है कि अभी हम लोगों को ये जो नई टेक्नोलॉजी है, इसमें थोड़ा टाइम लगता है. अभी वो वाली बात नहीं है जो क्लासरूम की टीचिंग में होती है. अभी शुरू में तो जब ये पिछले साल जब शुरू हुआ ये कोविड के में ऑनलाइन, तो मुझे तो कुछ समझ ही नहीं आ रहा था कि मैं क्या करूं क्या ना करूं. ये जो है अपने वो बेटी से पूछता था कि ये कैसे करना है, क्या करना है, कहाँ कैसे कोई प्रॉब्लम आ जाती थी. अब जाके थोड़ा बहुत, लेकिन अभी वो बात नहीं है जो क्लासरूम में जो टीचिंग होती है. So ultimately the classroom teaching is, I'm glad कि अगस्त से इस महीने we are going to start from 20th of August. So
हेयर कॉलेज हमारे यहाँ खुल गए हैं, सब बच्चे आने शुरू हो गए हैं.
हमारे यहाँ भी आ गए अभी. ये असल में अभी ये डेस्कटॉप पे कर रहे हैं ये सारा. जी. तो डेस्कटॉप पे ये यूनिवर्सिटी का इंटरनेट अच्छा है, इसमें इंटरप्शन नहीं होता. घर वाले में हमेशा 5-5 2-2 1-1 सेकंड का इंटरप्शन होता रहता है. इसलिए मैंने कहा यहाँ पे ऑफिस आके अपना करूँगा.
बहुत अच्छा लगा डॉक्टर साहब आपके साथ.
थैंक यू वेरी मच सर. थैंक यू. नमस्ते. टाइम एनी लेक्चर. नमस्ते सभी को.
नमस्कार सर.
सभी को नमस्ते.